Good morning, good afternoon, and good evening to everyone who joins us from around the world. We're so glad you're here. This is Andrew and Wadey. Welcome to the Stack series. Today, we are exploring sex as a biological variable in medicine.
And to really get into this today, we are looking at a pretty sweeping collection of sources. We've got clinical trial histories, pharmacological data, detailed cardiovascular and neurological studies, and even some internal policy documents from the National Institutes of Health.
Yeah, the mission for our exploration today is ambitious, but I think it's so critical. We really need to unpack how biological sex and gender fundamentally change the way human bodies experience disease, how they process medication, and exactly how the global medical system is currently adapting to this reality.
It's a massive shift in how we understand healthcare.
Right, and before we really get going, I want to start by asking you, the listener, a very direct question. Have you ever considered that the recommended dose on your prescription bottle, the one you take without a second thought, might have been calculated for a body that functions entirely differently from yours?
That is, it's a profound question, and it perfectly sets the stage for where we need to begin. Because if we want to understand where medicine is heading, we have to establish our foundation.
What we think we know.
Exactly, or rather, what the medical establishment thought it knew for a very, very long time.
Let's examine that historical baseline, because it's wild. For decades, the entire field of medicine practically operated on this underlying assumption that women were essentially just small men.
Yeah.
Like when it came to clinical trials and drug testing, the standard default body was male. If a drug worked safely on a 170-pound man, the assumption was you could just scale down the dosage based on weight, give it to a 130-pound woman, and just expect the exact same physiological results.
Which is a massive oversimplification, and it's one that has had cascading effects on global health. But before we get into the biology of why scaling down dosage by weight is fundamentally flawed, it is really important to clarify our terms.
Right. Definitions matter here.
The language we use matters immensely when navigating this medical history. So according to the American Medical Association, there is a clear distinction between sex and gender. Sex refers strictly to the biological, the chromosomal, and physiological differences between males and females.
Okay. And gender?
Gender refers to the continuum of complex psychosocial self-perceptions, attitudes, and expectations.
And that distinction is key because the historical exclusion we are talking about today was based on biological sex. Women of childbearing age were routinely and systematically excluded from early-phase clinical trials for decades.
Yes, a lot of this stemmed from a well-intentioned desire to protect potentially pregnant women from adverse drug effects.
Right, but the result was this massive gap in modern healthcare regarding how drugs actually affect female biology. We essentially built a massive pharmaceutical infrastructure with only half the blueprint.
And to understand the severe consequences of missing that blueprint, we really have to look at how medications actually interact with the human body at a cellular level. Which brings us to two fundamental concepts in pharmacology.
Pharmacokinetics and pharmacodynamics.
Exactly. Or, as you'll often see them abbreviated in the literature, PK and PD.
So to make sure we're all on the same page, let's break down the mechanics of those two concepts. Pharmacokinetics PK is what your body does to the drug. It's the journey the pill takes.
Right, how you absorb it through your gut.
Yeah, how you distribute it through your bloodstream and tissues, how your organs metabolize it, and eventually how you excrete it.
And then pharmacodynamics PD is the flip side of that coin. It is what the drug does to your body. It's the therapeutic effect at the receptor level, the chemical changes it triggers, and, crucially, the side effects it causes.
The biology here is where sex differences become undeniable. Biological sex directly affects the Cytochrome P450 enzymes, often just called CYP450, as well as the transporter genes located in the human liver.
Right, and these enzymes are the primary mechanisms for drug metabolism. They are the chemical workhorses that break down the active ingredients in your medications.
It's kind of like, think of your liver as a massive, highly efficient chemical processing plant. The drugs you take are the raw materials coming into the plant. And the CYP450 enzymes, those are the conveyor belts moving the materials along the assembly line.
That's a great analogy. They dismantle the chemicals so your body can either use them or flush them out.
And what the data tells us is that these conveyor belts run at completely different speeds depending on your biological sex. Some belts run much faster in female bodies, while others run much faster in male bodies.
Exactly. If you put a raw material on a conveyor belt that is running slower than the factory engineers planned for, things are going to back up. the drug accumulates.
Which leads us directly into a major historical case study that illustrates what happens when that small men fallacy meets the reality of those CYP450 enzymes. I'm talking about zolpidem.
Which most of you probably know better by its brand name, Ambien.
Right. For years, millions of people had been taking this sleep aid based on standardized dosing recommendations. The initial clinical trials, which were heavily skewed toward male participants, determined a standard dose that seemed safe and effective.
But over time, the real-world data revealed a severe problem. Women metabolize zolpidem much more slowly than men.
Going back to the chemical plant analogy, the conveyor belt for this specific chemical compound was running at a crawl in female livers compared to male livers.
Yeah, the drug simply wasn't being broken down and cleared from the system during a normal 8-hour sleep cycle.
The result was that women were waking up the next morning with the drug still heavily active in their bloodstreams.
It led to a very dangerous risk of next morning impairment. People were getting behind the wheel of a car, driving to work without realizing their cognitive and motor functions were still severely compromised by a sedative they took the night before.
And the fallout was so significant that in January 2013, the FDA made an astounding, unprecedented announcement. They had to step in and fundamentally change the recommended dosing guidelines for zolpidem.
They couldn't just rely on a blanket milligram amount based on weight anymore. The new guidelines dictated that the dosage had to be specifically tailored based on age, biological sex, and hepatic function.
They officially cut the recommended dose for women in half.
Yes. This was a watershed moment because it was a public institutional acknowledgement that biological sex is a critical variable in how a chemical compound interacts with human cells.
It is a perfect example of why this matters for you, the listener. It isn't just a theoretical debate for academics, but this doesn't stop at how we process synthetic drugs. It extends to the fundamental physical structure of our organs.
Absolutely. Let's look at baseline cardiovascular anatomy. The structural differences in the human heart between sexes are profound, even before any disease is present.
What does the clinical data show?
It outlines how women have distinctly different anatomical and physiologic baseline features compared to men. For instance, women generally have less left ventricular mass. The walls of the heart's main pumping chamber are physically less massive.
I want to pause on that because it's a critical structural difference. If the left ventricle is the primary engine pushing oxygenated blood out to the entire body, and that engine is physically smaller or less massive in women, how does that affect the day-to-day operation of the cardiovascular system?
Well, it changes the baseline hemodynamics entirely. Because of that structural difference, women generally operate with lower baseline blood pressure compared to men. However, to compensate and maintain adequate cardiac output, female hearts typically have greater contractility.
Meaning the heart muscle squeezes with a different, more vigorous dynamic force with each beat.
Correct. Furthermore, there is less cell turnover in the female heart compared to the male heart.
The actual cells.
Yeah, the cardiomyocytes, the muscle cells of the heart, aren't replaced at the same rate. These baseline differences set the stage for how diseases develop, how they progress, and why they present so differently when things go wrong.
Since we're already looking at the physical differences in the heart structure and mechanics, how does that translate when the system starts to fail? Let's move into the active debate within the medical community.
This is where things get really intense. Scientists are working aggressively to understand the diverging pathophysiological pathways of diseases in men versus women.
And there is no better example of this active, high-stakes debate than heart failure. Heart failure isn't just a single event like a heart attack, right? It's a chronic, progressive condition where the heart can't pump enough blood to meet the body's needs.
And the numbers here are staggering. By 2030, the prevalence of heart failure in the U.S. alone is predicted to rise to 8.5 million people.
To understand the medical debate, you need to be familiar with the two main types of heart failure, or HF. First, you have heart failure with preserved ejection fraction, known in the medical community as HFpEF.
Right.
Second, you have heart failure with reduced ejection fraction, known as HFrEF.
And just for clarity, ejection fraction is simply the percentage of blood leaving your heart each time it contracts. A normal ejection fraction is usually between 50 and 70%.
A helpful way researchers are framing this to differentiate the two phenotypes is by looking at HFrEF, reduced ejection fraction, as a plumbing issue.
A plumbing issue, exactly.
The pipes feeding the heart are blocked, there's a lack of blood flow, or there is direct physical damage to the pump itself, perhaps from a previous heart attack. The heart muscle becomes weakened, dilated, and just can't squeeze hard enough to push the blood out. The ejection fraction drops significantly.
Conversely, think of HFpEF preserved ejection fraction as a problem of stiff walls.
Stiff walls.
Yes. The plumbing might be relatively clear, and the heart might actually be squeezing out a normal percentage of the blood that is in the chamber, hence the term preserved ejection fraction. But the walls of the heart have become so thick and stiff that the chamber can't relax and fill properly with blood in the first place.
So even if it's pumping a normal percentage, the absolute volume is too low.
Exactly. If the chamber doesn't fill adequately, even a normal percentage of ejection isn't enough to sustain the body.
Now, here is the sex divide that the medical field is intensely debating. Men are heavily predisposed to the plumbing issues. Macrovascular coronary artery disease, where large arteries get clogged with plaque, and myocardial fibrosis. This structural pathway leads them primarily down the road to HFrEF.
But women have a significantly higher lifetime risk of developing the stiff walls HFpEF.
The debate centers on why these two phenotypes diverge so sharply along sex lines.
Right. For women, the pathophysiology of HFpEF is strongly linked to coronary microvascular dysfunction. Instead of the large arteries getting blocked, it's the tiny microscopic blood vessels in the heart that stop functioning correctly.
And this is coupled with severe endothelial inflammation, right?
Yes, which is chronic inflammation of the inner lining of the blood vessels.
Let's look at the specific risk factors driving this. We have data from a fascinating retrospective cohort study of 11,193 patients that details the unique and non-traditional risk factors for these different heart failure phenotypes.
When researchers looked at the women who develop HFpEF, the findings were quite surprising. The data showed that HFpEF in women is strongly linked to older age, an elevated body mass index, or BMI, and most unexpectedly, osteoarthritis.
Wait, osteoarthritis. How does joint degradation and cartilage loss in your knees or hips translate to stiff walls in your heart? That seems like a massive leap to make.
It does seem disconnected until you look at the systemic nature of the human body. The prevailing hypothesis in the literature is that osteoarthritis isn't just a localized structural problem, it leads to chronic systemic inflammation.
Because the immune system is responding to the joint damage.
Exactly. When your joints are constantly inflamed, your immune system is continually releasing inflammatory cytokines into your bloodstream. This chronic inflammatory state contributes to metabolic syndrome.
So the constant circulation of these inflammatory markers exacerbates the very endothelial dysfunction and fibrosis that cause those stiff walls on the heart muscle.
It's a chain reaction. The study also noted the specific role of visceral adipose tissue, the metabolically active fat stored deep inside the belly around the organs.
Right.
High levels of this specific type of fat contribute significantly to severe hemodynamic abnormalities, essentially creating abnormal blood flow pressures that force the heart to work harder, particularly during exercise.
Now, we also have to look at the other side of the coin. Even though women are more prone to HFpEF, they still develop HFrEF, the plumbing issue. And the cohort study highlighted a very strong specific link here. HFrEF in women is heavily linked to the Black race.
When you look at the recent multi-ethnic studies, the data on Black Americans is staggering. The research draws heavily on the multi-ethnic study of atherosclerosis, or MESA, and the atherosclerosis risk in communities study, known as ARIC.
Both of these massive long-term studies show incredibly high burdens of new-onset heart failure in Black Americans compared to other demographic groups.
The nuances here are critical for understanding the intersection of race and biological sex. The data shows that while Black men have the highest overall rates of HRDF, Black women have significantly higher rates than white women.
This points to a really complex interplay of genetic predispositions, systemic environmental factors, chronic stress, and biological sex that the medical community is still trying to fully map out.
And then we have the shared risk factors. The cohort study found that both types of heart failure in women, whether it's the stiff walls or the plumbing issues, are heavily linked to conditions you might not immediately associate with cardiology.
Specifically, asthma, depression, anxiety, and hypothyroidism.
The asthma detail is particularly striking. The clinical literature references a massive prospective study involving over 200,000 patients. It found that having a history of asthma is associated with a 2.14-fold increased hazard of developing heart failure later in life.
Over double the risk.
Yes. And crucial to our discussion, that association is significantly stronger in women than in men. Again, this points back to the theme of systemic inflammation.
Because asthma is fundamentally an inflammatory disease of the airways.
Exactly. That chronic inflammatory burden takes a toll on the cardiovascular system over decades.
The mental health details are equally critical here. The data emphasizes that depression and anxiety in women with heart failure are not just secondary emotional responses to being chronically ill, they are physiological risk factors.
They are linked to much higher risks of cardiovascular death. Yet, despite this severe risk, depression and anxiety are frequently underdiagnosed and undertreated in cardiovascular clinical settings.
This forces us to ask, what is the biological glue connecting all these disparate inflammatory, metabolic, and vascular issues, specifically in women?
The endocrinological experts point heavily to the estrogen factor.
Let's unpack that, because estrogen isn't just about reproduction.
Not at all. The dominant hypothesis is that estradiol, a primary, highly active form of estrogen, acts as a master regulator of inflammatory factors and vascular health in the female body. It helps keep blood vessels flexible and regulates lipid profiles.
But when estradiol levels naturally and precipitously decline during menopause, that protective regulatory shield drops.
This leads to a cascade of physiological changes. Without that estradiol regulation, blood pressure shifts upward, lipid content in the blood alters unfavorably, and body fat redistributes, often increasing that dangerous visceral adipose tissue we mentioned earlier.
All of these postmenopausal changes are direct, compounded risk factors for developing HFpEF.
It paints a picture of the body as a highly interconnected ecosystem. But cardiology isn't the only area where we see a massive active debate regarding sex disparities. We also have to look at the entirely different, highly subjective realm of pain management.
This is a deeply entrenched issue. There are numerous scoping reviews regarding gender bias in the management of musculoskeletal pain. The active debate here isn't necessarily about the cellular pathways of nociception, how pain signals travel to the brain, but rather how the healthcare system and the human beings working within it interact with patients experiencing that pain.
The core issue is how healthcare providers assess, validate, and manage pain differently between the sexes. The literature consistently shows that these differing assessments often lead to stark disparities in treatment.
For example, the data shows a man presenting with severe musculoskeletal pain is statistically more likely to receive aggressive pain management, including opioid analgesics.
Whereas a woman presenting with the exact same symptoms is more likely to be prescribed mild analgesics or have her pain attributed to psychological factors like anxiety.
It is an area of medicine where the subjective interpretation by a provider collides with biological reality. The scientific community is aggressively debating how to standardize pain assessment protocols to correct this implicit bias, essentially ensuring that treatment is based on the physiological reality of the patient, rather than preconceived gender norms.
Understanding these active debates is crucial, but it also pushes us to look forward. This brings us to the frontiers.
The frontiers.
Yes, the cutting-edge research at the intersection of sex, genetics, epigenetics, and neurology that might literally change everything we know about how diseases develop.
And there is no better place to start exploring those frontiers than the human brain. Let's look at what researchers are calling the Alzheimer's triad: age, APOE, and sex. You might have heard of Alzheimer's disease and the APOE4 allele in the news, but the way this specific genetic marker interacts with biological sex is absolutely fascinating.
To give you context, APOE4 is a genetic risk variant. In modern genetics, researchers use tools called polygenic risk scores.
Rather than looking for a single disease gene, right?
Exactly. They look at a vast array of your genes and calculate a cumulative score that predicts your susceptibility to a complex disease over your lifetime. In these predictive models, carrying the APOE4 allele is a major red flag for Alzheimer's disease.
But the critical insight here is the sex-dependent effect. Having the APOE4 allele doesn't impact men and women equally. The data clearly shows that the presence of this specific allele makes women significantly more susceptible to Alzheimer's and other age-related neurodegenerative disorders than men who carry the exact same genetic variant.
It's a profound discrepancy, and the evidence for this isn't just statistical data pulled from population surveys. It is physically visible in the anatomy of the brain. The clinical literature outlines the existence of sex-specific preclinical Alzheimer's neuroimaging biomarkers.
Meaning that long before a patient forgets a name or misplaces their keys or shows any cognitive decline, long before any behavioral symptoms appear, we can actually see the different risk profiles manifesting structurally in brain scans.
Correct. We can observe differences in brain volume, glucose metabolism, and amyloid plaque deposition. And those early physiological manifestations look distinctly different in a female brain carrying the APOE4 allele versus a male brain carrying the same allele.
That's incredible.
And this sex and APOE4 interaction isn't isolated to Alzheimer's. It also heavily affects the risk and progression of dementia in Parkinson's disease.
It forces a fundamental biological question.
Yeah.
How do the exact same genetic codes produce entirely different physical realities depending on biological sex? To answer that, we have to delve into epigenetics.
Epigenetics is arguably one of the most exciting frontiers in modern biology. It's the study of how your behaviors and environment can cause changes that affect the way your genes work.
A great way to visualize epigenetics is thinking about a computer. If your DNA is the hardware that you were born with, the physical circuits, the hard drive, the permanent infrastructure, then epigenetics is the software.
I like that.
It is the operating system that determines which programs actually get to run, when they run, and how loud the volume is turned up on certain genetic expressions.
From a biochemical standpoint, this software operates at the level of chromatin. Chromatin is the highly organized complex of DNA and proteins, primarily histones, packed inside the nucleus of your cells.
And the epigenetic mechanisms dictate how that works.
Right, mechanisms like DNA methylation or histone modification dictate how tightly wrapped or how accessible that DNA is to the cellular machinery that reads it. If the chromatin is tightly coiled, the gene is silenced. If it's unwound and accessible, the gene is expressed.
And the breakthrough finding in the neurological research is that chromatin accessibility fundamentally differs between the sexes in various regions of the brain.
Yes, and we have to ask what drives that differing accessibility. The answer points heavily to hormones. Specifically, gonadal hormones have what researchers call activational effects during adulthood.
Think about the physiological changes that occur over the course of the ovarian hormone cycle. These aren't just systemic changes affecting reproductive organs. The fluctuating levels of estrogen and progesterone lead to transient, measurable changes in the actual physical structure and function of the brain.
The hormones are essentially rewriting the epigenetic software on the fly, winding and unwinding the chromatin to express different genes at different times in the cycle.
This is a revolutionary concept for understanding brain health. It directly underpins the sex differences we see in susceptibility to a wide range of neurological and psychiatric diseases. The brain's software is constantly being updated and modified by hormonal inputs, and those inputs are profoundly sex-dependent.
Let's shift from the brain to the immune system, because the frontiers of autoimmunity are equally wild. We've known for a long time that women are disproportionately affected by autoimmune diseases. Around 80% of all patients diagnosed with autoimmune diseases are female.
But now researchers are finally mapping the exact molecular mechanisms driving this massive disparity.
The clinical data breaks down a specific immunologic mechanism known as the toll-like receptor 7, or TLR7, pathway. This pathway is a major driving force behind the massive sex bias we see in systemic lupus erythematosus, commonly known as SLE or simply lupus.
To put it simply for you, this TLR7 pathway is part of your innate immune system. It acts like an internal security alarm inside your cells, designed to detect the genetic material of invading viruses.
When it detects a threat, it triggers a massive inflammatory response to fight the infection.
But in lupus, that alarm system becomes faulty. It causes the immune system to essentially misidentify the body's own harmless cellular materials as dangerous viral invaders, leading the body to attack its own tissues.
The frontier discovery here is that this specific alarm system operates fundamentally differently in females than it does in males. The gene that encodes for the TLR7 receptor is located on the X chromosome.
Genetics.
Exactly. Because females have 2 X chromosomes, they generally have a higher baseline expression of this receptor compared to males who only have 1. Even though one X chromosome is typically inactivated in female cells, some genes, like TLR7, can escape that inactivation.
So females literally have more of these security alarms wired into their cells, making the system much more prone to misfiring and triggering an autoimmune attack.
Exactly. Understanding that the TLR7 pathway functions differently based on biological and chromosomal sex opens entirely new frontiers for targeted therapies.
Instead of using a broad sledgehammer approach to suppress a patient's entire immune system, which leaves them vulnerable to infections, researchers can now look at designing highly specific drugs that target the female-specific overexpression and operation of this exact pathway.
So we've looked at the history of clinical trials, the deep cardiovascular debates, and the cutting-edge science of epigenetics and immunology. But science doesn't happen in a vacuum.
It requires immense amounts of money, stringent rules, and massive institutions. We have to examine the implications of all this data. How is the scientific and governmental establishment actually trying to fix these historical blind spots and physiological disparities?
We have to look at the ethics, the economics, and the staggering bureaucracy of the National Institutes of Health.
The NIH has recognized these historical failures and the overwhelming biological evidence we've been discussing. In response, they implemented a crucial, sweeping policy mandate known as SABV. SABV stands for sex as a biological variable.
Let's walk through what this rule actually means for a bench scientist working in a lab today. Under the SABV mandate, researchers applying for federal funding can't just default to studying male mice or male cell lines simply because it's cheaper or because it's the way their lab has always done things.
They must conduct comprehensive literature reviews of both human clinical data and preclinical animal data to understand how sex might influence their specific area of study.
And here is the regulatory teeth of the SABV policy. If a scientist says, well, there is no previous data on females regarding this specific cellular pathway, the NIH explicitly states that the absence of data is not a justification to study only one sex.
You can't use a historical blind spot as an excuse to keep your eyes closed. If you want to propose a single-sex study today, you must provide an incredibly strong, specific, and scientifically rigorous justification for doing so.
Perhaps you are studying a disease that strictly affects the prostate or the ovaries, but short of that, you must include both sexes.
To guide researchers in this new paradigm, the NIH promotes what they call the 4 C's of studying sex to strengthen science. Researchers are instructed to consider sex in their experimental design, collect data on both sexes, characterize the differences they find, and communicate their findings clearly in their publications, even if they don't find a significant difference.
But changing the rules on paper is only one part of the equation. You have to understand the scale of the bureaucracy involved in science funding to see why change happens so slowly. The NIH is the largest biomedical research sponsor in the entire world. We are talking about an annual budget of approximately $48 billion.
Navigating that $48 billion is an absolute gauntlet for researchers. It is a highly competitive, incredibly stressful process. You don't just ask the NIH for money to study heart failure or lupus. You start by finding a program official, a PO, at a specific institute or center known as an IC within the NIH umbrella.
The NIH is made up of 27 different institutes and centers, each with their own specific focus and budget. Once you find your IC and your program official, you submit a grant application. And this isn't a brief proposal.
These applications routinely exceed 100 pages of dense, highly technical scientific protocol, preliminary data, and budget justifications. That massive application goes to the Center for Scientific Review, or CSR.
From the CSR, it gets handed to a scientific review group, an SRG, also commonly referred to as a study section. These are panels of peer reviewers, other scientists in your field.
They read the 100-plus pages and assign impact scores ranging from 10, which indicates a high-impact, nearly flawless proposal, to 90, which is low impact. Those raw scores are then converted into percentiles to determine who actually gets the limited pool of money.
Now, if we connect this massive bureaucratic structure to the bigger picture of women's health, we see how the system itself has inadvertently created and sustained disparities. The structural fragmentation of the NIH directly impacts what gets funded and what gets ignored.
The clinical literature gives a very real-world example of this structural problem regarding 3 interconnected conditions: uterine fibroids, endometriosis, and pelvic floor disorders.
All 3 of these are massive, critical women's health issues that cause chronic pain and affect millions of women globally. But because of how the 27 institutes of the NIH are siloed, research for these interconnected issues gets severely fragmented.
A grant application to study the cellular mechanisms of endometriosis might get routed to NICHD, the National Institute of Child Health and Human Development.
But if a researcher submits a grant to study pelvic floor disorders, that application might get sent to an entirely different institute, NIDDK, the National Institute of Diabetes and Digestive and Kidney Diseases.
This fragmentation means that these critical, often overlapping women's health issues are competing in entirely different funding pools, reviewed by different committees with different scientific priorities and different budgets.
The end result of this fragmentation is measurable. The data clearly shows that this siloed approach leads to chronically low levels of overall grant funding and massive persistent research gaps in these specific areas of women's health compared to diseases that have a dedicated centralized institute.
It's a systemic bottleneck. But the NIH is actively attempting to fix the machinery of how science is funded. To combat systemic bias and what they term reviewer fatigue, the sheer exhaustion of peer reviewers trying to grade 100-page grants across dozens of metrics, the NIH is rolling out a massive change to how it reviews research project grants, or RPGs.
This overhaul is starting in 2025 and is known as the Simplified Framework. Currently, peer reviewers have to score applications across 5 distinct, heavily weighted criteria: significance, investigators, innovation, approach, and environment.
Under the new 2025 framework, they are consolidating those 5 criteria down to just 3 broader factors.
This might sound like insider baseball to anyone outside of academia, but it is a monumental shift in how scientific futures are decided. A primary goal of this simplification is to address deep, long-standing criticisms that an applicant's past reputation or the prestige of their university was heavily biasing the outcomes under the old investigators and environment criteria, rather than reviewers focusing purely on the actual science being proposed.
Furthermore, by simplifying the scoring framework, the hope is that peer reviewers will have the mental bandwidth and the structural mandate to better assess an application's strict compliance with vital modern policies, specifically the SEBV mandate we just discussed. It forces the system to prioritize rigorous, sex-inclusive science over institutional prestige.
All of this funding, all of this cellular research, all of these bureaucratic overhauls ultimately scale up to a global level. We can tie this entire exploration together by looking at the recent massive global health study published in The Lancet Public Health.
The Lancet study looked at the global burden of disease across multiple decades and across different regions of the world. And the big picture conclusion is undeniable.
When you look at the major causes of disease burden worldwide, from cardiovascular disease to mental health disorders to autoimmune conditions, there are stark, undeniable differences between females and males in both prevalence and severity.
The ultimate implication for our society, for our global economics, and for the future of medicine is that we are in desperate, immediate need of gender-responsive approaches to global health. We simply cannot afford to treat the global population with a one-size-fits-all medical paradigm when the biological reality is fundamentally binary in its response to disease.
The health and longevity of half the global population depend on the medical establishment recognizing these differences at the cellular level, implementing them at the clinical level, and funding them at the policy level.
Which brings us to a final provocative thought for you to ponder long after we finish today. Consider the medications sitting in your bathroom cabinet right now. The pain relievers, the allergy meds, the prescriptions.
If every single drug currently on the market were forced to be reevaluated today with strict sex-specific and genetic parameters, how much of our established medical rulebook would have to be entirely rewritten?
It's a question that challenges the foundation of everything we thought we knew about pharmacology. Before we wrap up, thank you to all our partners, sponsors, and scholars who made this podcast and the research possible. We're truly grateful for your support. And to our listeners, thank you for being part of this journey with us. We hope you enjoyed today's discussion. If you did, be sure to subscribe and support us on Patreon. And of course, you can write to us in comments if a specific topic needed to be covered, which help us all to grow.
Thank you all so much for listening.
Until next time, stay curious, keep learning, and let the search for wisdom guide you forward. Have a great day.